Hi there. As always, I'm grateful to you for tuning in. Thanks. This is Professor Jared Rathel, and in this presentation, I'm going to be discussing the evolution of the larger, more complex eukaryotic cell. Of course, the protists, the plants, the fungi, and the animals, like you and I, are all composed of eukaryotic cells. So we have very strong evidence for how the eukaryotic cell evolved through these ancient mutualistic relationships between prokaryotes in a process called endosymbiosis. The first question I want you to think about is what actually limits the size of cells? So I'm composed of about 30 trillion eukaryotic cells and also about 39 trillion prokaryotic cells. Why can't I just be one gigantic cell? One single-celled big blob of a Rathel on your screen? The answer is cells are limited by their surface area to volume ratio. Small cells, like you see on the left here, have more parts surface relative to their volume than larger cells, like you see here on the right. Now, obviously, cells are not cube-shaped, but cubes make it easy to demonstrate this basic physical property. So a cube that has sides that are 1 millimeter in length means that one surface is equal to base times height, 1 times 1, or 1 millimeter squared. There, of course, are 6 surfaces on a cube, so the total surface area for this smaller cube on the left is 6 millimeters squared. The volume is base times height times depth, 1 times 1 times 1, 1 millimeter cubed. So what that means is we have a 6-part surface to 1-part volume surface area ratio. Okay, now let's grow that cube to have sides that are 4 millimeters in length. 4 times 4 gives us a surface area of 16 millimeters squared for one surface. 16 times 6 gives us 96 millimeters. So the surface area has grown as the length of the side has grown, as we would expect. But the surface has not grown as fast as the volume. The volume, of course, is 4 times 4 times 4, which yields 64 millimeters. 96 to 64 is equal to 1.5 parts surface to 1 part volume. So cells have to have a high surface area to volume ratio because they rely on diffusion, the movement of molecules from areas of high to low concentration. So they need to get oxygen in order to do cellular respiration, and they need to rid themselves of the waste product, carbon dioxide. I cannot consist of one giant Rathel cell because I would not have enough surface to get oxygen distributed throughout my single-celled body. So remember in the short video that you just watched, the time that oxygen almost killed everything, 2.1 billion years ago, the Huronian glaciation that covered the entire Earth in glaciers has ended and the planet is warming. Oxygen has filled the atmosphere, and we now have this protective layer of ozone, making the Earth much more hospitable to life, at least life as we know it. So all that O2 in the atmosphere is now going to allow cells to get physically larger. So they can get away with a smaller surface area to volume ratio because of the increased partial pressures of O2 in the atmosphere. And it's not coincidental that 2.1 billion year old rocks yield fossilized evidence of the first eukaryotic cells. So recall that prokaryotes are small. They're only about 1 to 5 microns in diameter. These fossil behemoths that you see before you are about 40 microns across. So that's like 8 to 40 times larger than the prokaryotes. Fair warning, you will see a similar illustration on your next assessment, and you will need to be able to identify which cell is a prokaryote, like you see on the left, and which cell is a eukaryote, like you do on the right. So while prokaryotic cells lack a membrane-bound nucleus and organelles, see on the left here, no nuclear envelope, the DNA is all floating around willy-nilly. Eukaryotic cells are larger and they possess a membrane-bound nucleus. Their DNA is now contained within a nuclear envelope, as well as membrane-bound organelles like the power plants of the cell, the mitochondria, and in autotrophic cells, the chloroplasts. So we're going to spend some time thinking about the origin of those mitochondria and chloroplasts today. So how do we make this giant evolutionary leap forward 2.1 billion years ago as prokaryotes evolve into eukaryotic cells? The evidence suggests it occurred through a process known as endosymbiosis.
What if you could absorb another organism and take on its abilities? Imagine you swallowed a small bird and suddenly gained the ability to fly. Or if you engulfed a cobra and were then able to spit poisonous venom from your teeth. Throughout the history of life, specifically during the evolution of complex eukaryotic cells, things like this happened all the time. One organism absorbed another, and they united to become a new organism with the combined abilities of both. We think that around 2 billion years ago, the only living organisms on Earth were prokaryotes, single-celled organisms lacking membrane-bound organelles. Let's look closely at just 3 of them. One was a big, simple, blob-like cell with the ability to absorb things by wrapping its cell membrane around them. Another was a bacterial cell that converted solar energy into sugar molecules through photosynthesis. A third used oxygen gas to break down materials like sugar and release its energy into a form useful for life activities. The blob cells would occasionally absorb the little photosynthetic bacteria. These bacteria then lived inside the blob and divided like they always had, but their existence became linked. If you stumbled upon this living arrangement, you might just think that the whole thing was one organism, that the green photosynthetic bacteria were just a part of the blob that performed one of its life functions, just like your heart is a part of you that performs the function of pumping your blood. This process of cells living together is called endosymbiosis, one organism living inside another. But the endosymbiosis didn't stop there. What would happen if the other bacteria moved in too? Now the cells, they were big and full of intricate structures that we call chloroplasts and mitochondria. These structures worked together to harness sunlight, make sugar, and break down that sugar using the oxygen that right around this time started to appear in the Earth's atmosphere. Organisms absorbing other organisms was one way species adapted to the changing environmental conditions of their surroundings. This little story highlights what biologists call the endosymbiotic theory, the current best explanation of how complex cells evolved. There's a lot of evidence that supports this theory, but let's look at 3 main pieces. First, the chloroplasts and mitochondria in our cells multiply the very same way as those ancient bacteria, which are still around, by the way. In fact, if you destroy these structures in a cell, no new ones will appear. The cell can't make them. They can only make more of themselves. Second piece of evidence. Chloroplasts and mitochondria both contain their own DNA and ribosomes. Their DNA has a circular structure that is strikingly similar to the DNA of the ancient bacteria, and it also contains many similar genes. The ribosomes, or protein assembly machines of chloroplasts and mitochondria, also have the same structure as ribosomes of ancient bacteria, but are different from the ribosomes hanging around the rest of the eukaryotic cell. Lastly, think about the membranes involved in the engulfing process. Chloroplasts and mitochondria both have two membranes surrounding them, an inner and outer membrane. Their inner membrane contains some particular lipids and proteins that are not present in the outer membrane. Why is that significant? Because their outer membrane used to belong to the blob cell. When they were engulfed in the endosymbiosis process, they got wrapped up in that membrane and kept their own as their inner one. Surely enough, those same lipids and proteins are found on the membranes of the ancient bacteria. Biologists now use this theory to explain the origin of the vast variety of eukaryotic organisms. Take the green algae that grow on the walls of swimming pools. A larger eukaryotic cell with spinning tail structures, or flagella, at some point absorbed algae like these to form what we now call euglena. Euglena can perform photosynthesis, break down sugar using oxygen, and swim around pond water. And as the theory would predict, the chloroplasts in these euglena have 3 membranes, since they had 2 before being engulfed. The absorbing process of endosymbiotic theory allowed organisms to combine powerful abilities to become better adapted to life on Earth. The results were species capable of much more than when they were separate organisms. And this was an evolutionary leap that led to the microorganisms, plants, and animals we observe on the planet today.
If I could engulf a cobra and spit venom, I would totally do that. So, the evidence for the endosymbiotic theory, the theory of endosymbiosis, it's quite compelling. To aid students in remembering this evidence, I always encourage them to remember the acronym, the MAD Doctor, like you see on the left, the MAD Doctor, where M stands for membranes. So both the mitochondria and the chloroplasts have double membranes. An outer membrane that's structurally similar to the larger eukaryotic cell within which they now live, as if they passed through this membrane a long time ago, as well as a second membrane, an inner membrane, that is structurally the same as free-living prokaryotes. Second line of evidence, antibiotics. If you treat certain algae cells, which are eukaryotic, with certain antibiotics, the antibiotics actually destroys their internal chloroplasts, as if those chloroplasts were free-living prokaryotes. The larger eukaryotic cell can't make more chloroplasts once they're gone. It doesn't have the DNA for them. So the larger cell eventually starves. The first D stands for division. Mitochondria and chloroplasts, they have their own independent cell cycle. They are dividing via binary fission, just like the prokaryotes do, on their own timetable, irrespective of what the larger cell is doing. The second D, which is quite convincing, stands for DNA. So both mitochondria and chloroplasts, they have their own DNA that's separate from the DNA we find in the nucleus of the eukaryotic cell. And the DNA in the mitochondria and the chloroplasts, it's circular, just like the DNA that we find in free-living prokaryotes. So, interesting tidbit, side note, you inherited your mitochondrial DNA, the DNA that's in all of your mitochondria in your 30 trillion eukaryotic cells. You got that DNA from your mother. And so there's been some really cool science done that's mapped mitochondrial DNA mutations, and it's proved this really powerful tool to help map out how humans colonize the planet. Check it out, it's called the Human Genographic Project. Lastly, both mitochondria and chloroplasts, they have their own ribosomes. And you remember a ribosome, that's the little cellular machinery that manufactures proteins. So the ribosomes found in mitochondria and chloroplasts are identical to the ribosomes that are found in the free-living prokaryotes, both structurally as well as their size. So I ask you to take just a moment and let this idea of endosymbiosis really sink in. What it implies is that eukaryotic organisms on the planet today, the protists, the plants, the fungi, and the animals like us, we're made of chimera cells. Each of our cells is really just a mishmash of bacterial cells working together in an ancient mutualistic relationship. So just like in Greek mythology, you remember the chimera? It's this, hodgepodge of a lion, a goat, and a serpent tail. Similarly, we're really just a mishmash of bacteria. So note here that the first eukaryotic cells were not anywhere close to plants, fungi, or animals. The first eukaryotic cells were composed of only one cell. They are unicellular. Multicellularity, it's not going to enter the stage for another billion years. And we'll talk about the evolution of multicellularity in our next lecture. Okay, so now really quickly, we're going to take a tour of the major lineages of the kingdom Protista. So the Protists aren't a monophyletic kingdom. They're really just this big grab bag of organisms that don't fit into the kingdom Plantae, fungi, or Animaliae. All Protists, all of them, are made from eukaryotic cells. Most protists are unicellular, although there are several multicellular lineages, like the brown algae that you see right here. You probably know this as kelp if you've ever been swimming off the California coast. Some protists are photosynthetic. Others have to acquire their energy from consuming other organisms, meaning they're heterotrophic. Some, like these euglena with their long whip-like flagella and really cool light, dark, sensitive eye spots, right? These guys have chloroplasts, but they also ingest prey, the euglena. Moving on, the diatoms. These guys are unicellular photosynthetic microalgae. Our oceans and freshwater bodies are teeming with these protists. In fact, they generate some 20 to 50% of the oxygen that you're breathing right now in the atmosphere. So the diatoms are beautiful. They're often referred to as the jewels of the sea, if you will, because they have these cell walls made of silica. And you can see these beautiful snowflake-like patterns. So the shells of dead diatoms can reach as much as a half-mile deep on the ocean floor. That's how many of these things there are. And actually, a cool factoid, the entire Amazon basin, the great tropical rainforests of the Amazon, those are fertilized by 27 million tons of diatom shell dust that's transported by transatlantic winds from the African Sahara. Cool factoids for your party on Friday night. Oh, how I love the dinoflagellates. So these are single-celled eukaryotes. Most are marine plankton that are photosynthetic, although some do ingest prey. In high concentration, these dinoflagellates actually cause red tides, which can be really nasty, causing fish to go belly up and wash up on the shore. But the truly mind-blowing characteristic about the dinoflagellates is their ability to bioluminesce. Bioluminescence is when living organisms are able to produce and emit light. So years ago, I experienced bioluminescent dinoflagellates. I was kayaking in Southeast Alaska. It was amazing. Every time you put the paddle in the water and disturbed the water, there were streaks of light. But let me just show you. So here is bioluminescent surfing just over in Oceanside, California in 2011. The ciliates are a large and diverse group of protists, including many that hopefully at some point you've had the chance to observe in a drop of pond water under magnification. So the more famous ciliates include the paramecium, like you see on the right, as well as stentor. So the ciliates are characterized by their cilia. These are those little tiny hair-like projections surrounding the organisms. That's my cilia dance. They use their cilia. Some do use their cilia for movement. Others are going to use those cilia to feed. And then finally, we have the cercozoans. The amorphous blob that you see on the right is a cercozoan known as an amoeba. So these guys feed by surrounding their prey with pseudopodia, their false feet, and then ingesting their victims. The amoebas are the wolves in the drop of pond water. So check out this amoeba putting the stalk on and consuming most of a ciliate called stentor.